Hello, everyone. My name is Michael Shortreed, and I'm a distinguished scientist at the University of Wisconsin-Madison in the Department of Chemistry. I work with Professor Lloyd Smith. It is my great honor to be able to have the opportunity to share some of the research of our group with you here today. Mass spectrometers can't identify peptides and proteoforms. No matter the format, they only provide lists of peaks, intensities, and times. The job of identifying peptides and proteoforms is left to the user. Without software, this task would be hopeless. I hope this sounds like an interesting premise for a talk, because that's what I'll be telling you about here today. It's my sincere belief that without software, this field that so many of us here are involved in wouldn't be anywhere near where it is today. At the start of the talk, I'll go over what I consider to be the dawn of modern proteomics, which is an experiment called peptide mass fingerprinting. I'll talk about some developments in software algorithms and approaches that took place in the 90s, and then move on to more current research. And then I'll end my talk with some things that are still remaining challenges in the field, challenges that are being addressed in large part through software development. Proteomics, by definition, is the large-scale study of proteomes. So certainly proteins were being identified prior to like 1993. But 1993 was the year first year when software was starting to be applied to the identification of many proteins from a single sample. And this was shown in an experiment called peptide mass fingerprinting. Here we've got the schematic on the left, where we've got a 2D gel-separated complex protein sample. Each band is excised in turn, and then proteins in that excised spot are digested with a protease. By measuring the intact mass of the peptides in that digested protein, you get a list of characteristic fragments. This was done either by fast atom bombardment or multi-mass spectrometry. And then you could compare this list of identified peptides to a list of theoretical peptides from a sequence database. And here on the right, you can see 3 different approaches that were published in that year using 3 different resources. The MOSE database, which is a DNA sequence database converted into proteins, the FragFit search algorithm, and the protein information resource. Of course, in the late 80s, electrospray ionization tandem mass spectrometry sort of was evolving, and eventually in 2002, Fenn and Tanaka got Nobel Prizes for that work. Here we can see the basic experiment, which is really still the same kind of experiment that's being performed today. Dig a complex sample of proteins, digest it with a protease into peptides. These are separated usually by reverse phase chromatography. Peptides behave really well by the separation mechanism, and it's easy to get very nice bands. Those bands of eluting peptides are measured by their intact mass, and then individual intact masses are isolated from fragmentation, producing a tandem mass spectrum. It wasn't really until 1994 when computer algorithms started to be able to interpret tandem mass spectrometry data. Here's the first published work that I found by Matthias Mann and Wilm in analytical chemistry. Here, they didn't really try to match the entire tandem mass spectrum to a theoretical mass spectrum, but they used different pieces of information to try to figure out what the best peptide match was using a sequence database. The first aspect of the experiment was to look for a small sequence tag using the most intense peaks. Here you can see 4 intense fragment peaks in the middle, and the first mass could be either leucine or isoleucine. The second one could be Q or K, even though they're not isobaric with the resolution and mass accuracy at the time. They didn't have the ability to distinguish those 2 amino acids. And then finally, the final mass is again either isoleucine or leucine. And so the candidate peptides to match this experimental mass spectrum would have had a sequence run of I or L, Q or K, and I or L. And so actually, so here's the correct assignment on the right, and you can see L, Q, I in the middle of the sequence. There was two other bits of information that were used in the identification. One was the mass from the beginning of the peptide to the smallest fragment. In this case, that matches to NSR, which is shown at the C-terminal end. That's a mass of 376 Thompsons. And then the other mass is the mass of the intact experimental mass minus the largest fragment. In this case, it's 1,023 Thompsons. And so using those two terminal fragment masses as well as the sequence tag was the way that they were used to identify the peptide. And you could do multiple proteins using their software called peptide search. Of course, everyone here is familiar with sequest. sequest was also published in 1994, and here what was done was simply matching theoretical mass spectra to experimental tandem mass spectra and scoring the match using cross correlation. I love these photos in the bottom left of Jimmy Yang, Ashley McCormick, and John Yates. I had no idea how old they were at the time, but they looked pretty darn young. So, interestingly, sequest and variations of sequest are still in widespread use today, which really speaks to the importance of that development at that time, almost 30 years ago. I didn't start in the field of proteomics until 2015. I've been in Professor Smith's lab since nearly the year 2000, so I spent 15 years there working as a surface analytical chemist, doing imaging, peptide and protein arrays, and so forth. And there were other people in my group doing mass spectrometry, but I wasn't one of them. And I think at 2015, I finally got on board with what I was doing, and the grass seemed greener on the other side of the fence. And so I decided to see if there was maybe something I could contribute. I didn't necessarily really know where to begin. Most of the people in our group were working on instrumental developments, and I didn't have any kind of ability in that area. But I did know how to program a little bit, and I did love, more than anything, doing data analysis. And so I began trying to do different searches of the mass spec data and finding multiple different PTMs in them. And one of the things that frustrated me most was having the people in my group tell me over and over again that you can't identify multiple different PTMs in the same sample. And the reason is that you pay a price in the false discovery rate. So I was left wondering about what could be done there. By some miracle, at that time, I was very lucky to be able to attend my very first ISMB conference. And it was such an exciting time seeing all the different talks and seeing all the different posters. One talk that I found really compelling was given by Professor Steve Gigi, where he was presenting work done by his student, Joel Chick. And in this experiment, instead of doing the typical bottom-up experiment where one matches experimental mass spectra with theoretical peptides having the same mass, he opened up the search and he simply let the experimental mass or experimental peptide match any theoretical peptide in the database regardless of its intact mass. And then reported the mass difference of the best matching theoretical peptide to the experimental peptide. Here you see the graph of the results that they got. There are these beautiful peaks rising out of the bottom, where along the x-axis we've got the mass difference between the observed and theoretical best peptide. On the top right, you can see a beautiful peak at 80 Thompsons, corresponding perfectly to phosphorylation. If you look closely, there are lots of other peaks, like for 42 Daltons, acetylation, 16 Daltons, oxidation, and so forth. And this was really inspiring to me because they had done what I had been wondering about, which is shown that one could see a variety of different PTMs in the same sample. Of course, this really wasn't a finished analytical product. It wasn't even a software program. It was just results from a search where they opened up the precursor mass tolerance. So I started wondering if there was a way that we could take this approach and turn it into a software program that other people could use to mine these PTM differences and allow people to have the ability to globally identify PTM modified peptides. To begin this work, we approached our friends from across the campus, student Craig Winger and Professor Josh Kuhn. While I could program a little bit, I really didn't have the ability to create my own search engine. And Craig had already done that, having already published a paper on a software program he called Morpheus. At that time, the paradigm had shifted from low-res to high-res mass spectrometry. And so one could take advantage of the much better resolution in MS1 and MS2 to do a much faster database searching with a much simpler algorithm that would be a lot faster than, really Sequest, which was like the #1 algorithm at that time. We talked to him about taking an approach where we did a open mass tolerant search. On the first round, we coded potential PTM candidates we observed by characteristic mass differences into a sequence database, and then followed that up with a second search to identify those PTMs. The way we recorded PTMs was using an XML entry in a sequence database. Here I'm showing you an example of one that I got from Uniprot. Most people use a FASTA database, which is a simple database that contains on one line the accession, the protein name, the gene, the organism, and then on the second line of an entry, the full protein sequence. What an XML entry has is additional information showing the location and specific types of various PTM modifications, the location of known signal and chain peptides, as well as the location of known amino acid variants. And so it's a much more comprehensive protein entry that can be used to look for various different features that one can't do simply using a FASTA sequence database. Well, that first publication, which we called GPTM for Global PTM Analysis, just wasn't very fast. The time it takes to do an open mass tolerant search is quite long, and most people aren't willing to sort of take the time for that. So we tried to come up with a way to really speed up the search, and that's when we came up with the concept for notch mass searching. So here, rather than allowing a experimental spectrum to match all theoretical peptides within 200 or 500 or 1,000 Daltons, we only allowed them to be matched to peptides whose theoretical mass was a specific mass difference away, plus or minus a few ppms in tolerance. This really increased the search speed by multiple orders of magnitude. It also surprisingly increased our sensitivity. And probably the most interesting thing was it also increased our peptide identification accuracy. So what we had found was when you totally left the search tolerance open, unrestricted, that you could get strange accidental matches between theoretical and experimental peptides just because of random fragment masses. And so this would leave you with a false positive identification or a decoy identification. However, when you restricted the search to certain notches, you couldn't be distracted from the correct match with an accidental false positive. And if you go and look at the annotated spectra, you could see that you got a more correct match, even though the score was slightly smaller. At this point, we really sort of felt like we had some beginning momentum in search software development, and Craig had left Josh Kuhn's group and was off doing his own thing, and we no longer could work very fast with him because we were coming up with all kinds of ideas and expecting him to integrate them into his software program. And we asked if he wouldn't mind if we forked his repository into our own and began using his software as the basis for a bunch of new developments. And he graciously said, go for it. So we named this quote unquote new software Metamorpheus so that people would always know that it came from Metamorpheus at its beginning. You can find all of our code for everything that we do available on GitHub. It's always available at every stage of the development. Currently, the Metamorpheus search software does calibration, search, PTM discovery, label-free quantification, cross-link peptide identification, and glycopeptide ID. It's available as a GUI or command line program, and it works in Windows, Linux, or Mac OS. I'm going to spend just a couple minutes talking about some of those features of Metamorpheus that are available, just to get some sort of an idea of what the kinds of things you can use, do in Metamorpheus. The first is label-free quantification. We have a student, Rob Milliken, who developed this program called FlashLFQ as a new student in our group. At the time he joined our group, Alexei Nezvishky had released the software program MS Fragger, and it was just breathtakingly fast. We were all captivated by how fast one could actually do a search. And even though we tried to implement those ideas into our own search program, we couldn't quite capture the same speed that his group was able to. But nonetheless, we were able to use that idea of doing lookup tables of masses to produce a very fast and accurate label-free quantification software program. Here you can see a couple of graphs from that publication. On the left, what we're showing here is 2-to-1 mixtures of E. coli spiked in a human protein background. And we're comparing the FlashLFQ quantification results to MaxQuant, which is really the top program for quantification at that time. And on the right, you can see a search or the quantification time comparison between MaxQuant in the gray bars and FlashLFQ in the small blue bars. It was more than an order of magnitude faster with equal or better sensitivity, precision, and accuracy. Another thing that you can do in Metamorpheus is a multi-protease search. So this is work that was done by Rachel Miller. People had already published papers where they used multiple proteases to improve the coverage of proteins, but no one had released a software program that could accomplish this feat. And so Metamorpheus was the first program that could do that. Here you basically load all your raw files and simply designate which protease to use with each one. And then during the search and match process, only peptides from the appropriate protease are matched to the experimental spectrum. We gather all the peptides before we do the protein inference. And in doing so, we can really reduce the protein ambiguity that you get compared with, say, trypsin alone. Another thing that's possible, because you can do PTM discovery in Metamorpheus, is that you can use multiple different proteolytic peptides that cover the same region of a protein to really localize PTMs. is simply, like much harder to do if you were to run separate searches for each protease and then try to combine all of the results together by hand. One thing Metamorphosis can do is glycopeptide identification. We have a search algorithm called OPAIR, which is integrated into Metamorphosis, and this was work that was co-created by Nick Riley, who was in Carolyn Bertozzi's lab at the time, and our graduate student, Lei Liu. Lei didn't have any prior experience working in this area, nor did I, but he was really excited about the opportunity to kind of work on this, and we are having really just good fortune to be able to work with Nick Riley, who is such an expert in glycopeptide analysis. It really helped us create a software program which is not only like highly usable, but also produces phenomenal results. In OPAIR, you can use paired CID and ETD dissociation spectrum for better identification and localization. We use just like in FlashLFQ, we use an ion-indexed open modification search approach to identify candidate O-glycosylations on the peptide. And then we use graph theory and probability-based localization to localize O-glycosites. This allows us to do up to 4 or 5 different O-glyco modifications on a single peptide. At the time we published this was before Alexei's group released their search. It was much faster than anything available either publicly or privately through commercial programs. One thing that we introduced in this paper was O-glycosate localization confidence levels. And that's kind of I think that's like an important thing for the field to think about because not all of these identifications are equivalent. Finally, in addition to several available glycan databases in OPAIR, users can use their own database. And so if they have interesting or custom glycosylations, they can search for those things in this software program as well. And finally, I just wanted to let you know that we also have a new spectral library search feature developed by graduate student Yuling Dai. This work has been submitted for publication, and we're in the process of revising the manuscript now. But here you can see this is actually a MetaDraw visualization software, which is part of the Metamorphosis package. And here, for every peptide, if it was matched against a library from a spectrum, you can see the experimental spectrum going from the middle up, and you see the best matching library spectrum in the mirror plot going from the middle down. And it allows you to kind of go through each spectrum and manually try to determine if this is a correct match or not, simply using the spectral library search. Now I'd like to spend a few minutes talking to you about proteoforms. Certainly, proteoforms are one thing our group is known for studying. In fact, the term proteoform was coined by Professors Neil Kelleher and Lloyd Smith some years ago, and it's really been adapted by the field as like the term of choice when talking about specific arrangements of amino acids and PTMs. We also came up with the concept of the proteoform family, which is all of the different proteoforms stemming from a single gene. These would include different splice variants, different amino acid substitutions, different PTMs, and so forth. So what's the deal with proteoform identification? Well, I try to include this slide in every talk that I give about proteoform analysis because I think it's really telling for the field and provides motivation for work here. In this graph, every gray dot represents a single protein measured by bottom-up proteomics. Proteins are identified by one or more unique peptides. Along the horizontal axis, we have the nominal protein mass, and on the vertical axis, we have the protein intensity. So you can see by bottom-up proteomics, we're seeing proteins that cover a wide mass and intensity range. Now, when we identify any proteoform that matches to a protein, we turn that dot from gray to pink. And so you can see a smattering of pink dots on the left portion of the graph. And there's very few of them. In fact, in this experiment, probably only 10% of the proteins had observable proteoforms by top-down mass spectrometry. You'll note that they're all lower proteins in mass and higher proteins in abundance. So this really is the state of top-down as we speak, and it clearly shows like how much more work has to be done in this area. So, like for us, it's like highly motivating in terms of our endeavors to do software development as well as to do different experimental technical developments. Of course, since now we have our own search software, Metamorpheus, we came up with a way to do top-down identification. This work is begun by Rob Milliken and then expanded on by Katie Henke. The way that we accomplished it was simply to turn top-down into a protease, basically a protease that didn't cut at any amino acid. And by taking this approach, all of the different facets of Metamorpheus were available to be immediately applied. That means that we can do calibration, we can do PTM discovery, we can discover truncated proteoforms, as well as identify signal and chain peptides. We can even find mutations. In work I'm not going to tell you about today, we have a way to generate a proteogenomic database by mining RNA sequencing data, and so that can be included into the search. Not all proteoform identifications are equivalent. The people that do proteoform analysis are very fond of publishing papers where the number of proteoforms identified is in the title. And we all like to compete with each other to get the biggest number in the title. But the fact of the matter is that when we're talking about identifying a proteoform, we don't necessarily all mean the same thing. If you think about it, we don't always know for a particular proteoform what all the PTMs are. Maybe we know what the PTMs are, but we don't know where they're located. Maybe we know a particular proteoform belongs to a protein, but some piece of it's been truncated and we don't know how much or what has been cleaved away. So we and others published a paper in Nature Methods with a 5-level classification scheme for proteoform identifications, beginning with level 1, where there's no ambiguity in terms of PTM localization, PTM identification, amino acid sequence, and gene of origin, all the way down to 5, where we basically have only a mass, and we know that it's a protein, but we don't know anything else about it. So in this way, by publishing the levels with each of our identifications, it gives people the opportunity to compare different approaches, different software approaches for peptide identification. And now, sort of basically having numbers, Metamorpheus for every identified proteoform provides a proteoform level. You can start to think about, well, how do we change the experiment to reduce the amount of ambiguity in those identifications? Now that we can measure ambiguity by counting proteoforms with the various classifications, what can we do to change that? Our student, Zach Rolfs, came up with a way of using internal fragment ions to disambiguate proteoform identifications. Here you can see a fragment map where B and Y ions are the blue and red bars, for a particular proteoform. And then the purple bars below that show internal fragment ions that were assigned to the various MS2 peaks observed. On the bottom right, you see a graph. This was for a complex proteoform analysis. We have all of the different proteoform levels as a different colored and numbered circle, with high ambiguity on the left, with the circle with the red circle 5, and low ambiguity on the right, with the blue circle 1. And then there are lines between each of the circles showing the increase or the decrease in ambiguity that was obtained by adding internal fragment ions to the annotation of the sequence. And so this was really a successful way for us to do a better job of proteoform identification, and it really required an innovative software development. It wasn't anything that was done in terms of the experiment itself. We've also done work in our group combining bottom-up and top-down data in a single search. Basically, since top-down really in Metamorphosis is treated just like a protease, and we can do multi-protease searches, that means that you can also include bottom-up peptides and top-down proteoforms in the same search result. So in the left there, you can see we've got various proteoform measurements in purple and bottom-up peptide measurements in red. And when you search them together, you can match up the peptides with the proteoforms and improve the PTM localization. On the right, you see as we integrate various pieces of information, starting on the left with only top-down XML from UniProt, and moving all the way to the right, combining bottom-up, top-down, and PTM discovery together, we see a vast increase in the number of PTMs, both identified as well as localized. One thing that we're really interested in is, since top-down experiments take such a long time and yield so few proteoform identifications, maybe we can simply identify proteoforms by intact mass alone. So this is work that was done by myself and Brian Fry several years ago. We made a SILAC mixture of proteins, heavy and light, and we mixed those things together in a defined ratio so that when you see a proteoform eluting, the MS1 has a characteristic ratio between fragments. You can measure the mass difference, the SILAC mass difference between those peaks, and compute the number of lysines in the proteoform. And then by looking up in a table of exact mass and lysine count, you can ID the protein. And we really think this is important is because one of the things that's holding back our sensitivity in top-down analysis is the speed of the mass spec experiment. And we're hoping that one day this might be an avenue towards accomplishing that. So I just kind of want to reiterate why proteoforms are so hard to identify. One is they can have any number of PTMs, like many PTMs, many different kinds of PTMs, many PTM combinations. They can have truncations, they can have splice variants, amino acid variants. So the thing about a proteoform is the tiniest variation changes the intact mass of the entire proteoform. And if we're trying to use a sequence database to look up that proteoform in, there's a good chance that it won't be there. In contrast to bottom up, where we take proteins and digest them into peptides, one or more peptides might have an unknown PTM. It might have an amino acid variant. There might be a splicing variant. But so long as the other peptides remain unchanged, we can map them to the correct protein and know that that protein is there. And so these changes don't really affect bottom up so much, but we have to weigh through experiments or software to improve the situation for proteoform identification. One thing that we're developing along this line is a new way of fragmenting proteoforms that improves the speed as well as the sensitivity of detection. Here we're doing a source-induced decay experiment, and this has been done for bottom-up. But basically, before the proteoforms even enter the mass spectrometer, the voltage is cranked up so that the proteoform starts falling apart. And so in comparison to a traditional experiment where you just isolate one charge state of a proteoform for fragmentation, here we're fragmenting them all together. To illustrate this, here's an example of a particular protein. And you can see the MS1 spectrum in the panel A, and there are so many different charge states of this protein G. If you pick charge state 24 in the purple box, you see the fragmentation in panel B. And if you pick the charge 14 envelope in the light blue panel C, you see a completely different fragmentation spectrum. And this is frequently true. Picking different charge states of the same proteoform gives a different fragmentation pattern. Not only does it reduce the amount of signal by choosing one of many charge states, it sometimes results in very, very poor fragmentation that won't result in an ID. In contrast with source-induced decay, we're fragmenting all of the charge states. So we can rapidly take a scan because there's so much signal coming in from all the different things. And then we see even fragmentation across the M/Z range, allowing us to do a much better job of mapping. So two things happen because of this. Because we're fragmenting so many ions, we can take MS1s a lot faster. So in the graph on the top right, in blue, you see the MS1s from the SAD experiment, and in green, the DDA experiment. And so there's a much, much higher density of MS1 spectra there. And then the bottom right, you can see a big increase from green to blue of proteoform spectrum matches, proteoforms and proteins identified in a complex sample when using the SID approach. Well, now it's 2022, 7 years since 2015. Is there still much to do? Yes. Now that I'm here and I'm not such a novice, I know there's so much more to do. There's lots of things that I'm wondering about, even though so many developments have occurred. I'm still wondering if PTMs matter. We've had GPTMD for years, and still people in biology aren't really taking advantage of it. They really are still doing enrichment type experiments for various, whether it's glycosylations or phosphorylations or acetylations. People aren't doing global PTM studies. One thing I'm wondering a lot about is if we can do proteoform inference from bottom-up data. Clearly, we have a lot more sensitivity in bottom-up, and I don't really see sensitivity in top-down changing all that much soon. So I know there's been some papers published along these lines, but I think we might need a nice software innovation that would allow us to infer proteoforms from bottom-up. One thing I wonder about is whether protein inference is good or bad. Biologists seem quite content just lumping all the peptides together by protein or even by gene and looking at differences. And I'm thinking so much information is lost when we do that. But maybe we need to take a closer look at that. I also want to know how much proteoforms actually matter. Even though those of us that study proteoforms are convinced, there's a lot of biologists in the world that really aren't going after proteoforms with as much excitement as we have. I wonder how much we can improve the sensitivity for proteoform identification purely with mass spectrometry, and thinking maybe there's a non-mass spec technology out there that will become the primary modality for proteoform analysis. As far as other challenges go, I've got some challenges for you. Try a new software program. Try ours, try someone else's, try anyone's. There's lots and lots of good work out there, and many of us have just tend to stick with what we know and what we love, and so I encourage you to give something new a try. If you do write code, make it available to other people. Put it on GitHub and let people see it and take advantage of it and build from where you left off. Just like we benefited from the work of Craig Wenger, I hope people will take our work and grow it. And I think it's really important for the field that we help each other out in this way. And also do what you can to maintain your code and respond to requests for help. We've met many great people taking this approach, and it's really, really nice when you try a new software program. If you find that it doesn't work or it doesn't do what you want it to do, you can get some help to do it. I'd like to close by thanking all the members of the group that worked in the computational biology area, and thank you for your attention.